Hello, all right. Chapter 13. No, yeah, chapter 13. One of the hottest topics now, viruses, right? So let's talk about all this cool stuff. All right. Scroll button. Share screen. Viruses. Okay, so viruses, viroids, and prions. We'll skip viroids. We'll focus on viruses and prions since they affect humans and not viroids don't affect us. They affect us, our crops, but not us directly. Okay, so, all right. Third objective, differentiate a virus from a bacterium. Okay, make sure you're able to do that. Okay, so first of all, a virus by definition is an obligate intracellular parasite, which means viruses on the outside of the cell don't do anything. They don't function, they don't have metabolism, they don't move, they don't do anything. Okay? They require living host cells to actually multiply. Okay? Viruses contain either DNA or RNA, okay? Never both, but either DNA and RNA, and they have strange forms of DNA and RNA, which we'll get into in a little bit. Okay, so like all living cells have DNA. Viruses can survive with just RNA. For example, the COVID-19 is an RNA virus. There's no DNA from it. Okay, so the whole thing about taking DNA from HIV and inserting it into COVID-19, and that's how they made it. As soon as I read that, I was like, oh, that's so stupid. Like, what type of moron would publish that? And you know, it turned out to be completely false. Okay, because COVID-19 doesn't have DNA. Okay. They also, by definition, contain a protein coat. Okay, so a virus has to have these two things: DNA, RNA, okay, and a protein coat around it. Okay, they have no ribosomes. They have no ATP generating mechanism, which means by definition they have no metabolism, right? And so, okay. So here's what viruses are. Okay. Okay. Also, most importantly, the sensitivity to antibiotics is a big no for viruses. So if a doctor tells you to take antibiotics for a common cold, you gotta find yourself a new doctor. Okay. All right. The host range. The host range is a spectrum of host cells a virus can infect. Okay. Most viruses infect only specific types of cells in one host. Okay, and it's determined by host-specific attachment sites and cellular factors. Okay. So back to COVID-19. If you guys have been paying attention to like exactly what COVID-19 is, COVID-19 has a big spike coming out of it called the spike protein. The spike protein attaches to a receptor called ACE2 receptor in humans. Okay, and that is their binding site, and that's their infection site. So what would happen if you have lower levels of ACE2? Well, the virus would find it harder to bind, and it would cause less illness. This is precisely the reason why little children do not get COVID-19 as much as adults do. And they definitely suffer from less because they literally have less ACE2 binding sites, and therefore less chance to get infected. And even if they are infected, they have less mechanism, like they don't cough nearly as much and stuff like that, and therefore they're not spreading it nearly as much. They know this is true for elementary school kids. They know it's not true for high school kids because in high school, by the time you hit high school, your level of ACE receptors raised to about adult levels, close to adult levels. They don't know about middle school. Okay, so that's where we're at now. Okay, and that report only came out 5-6 days ago, actually. Yeah. Well, you guys are watching this video in a couple of weeks, so maybe like 2 or 3 weeks ago. Okay. Bacteriophages are viruses that infect bacteria. Okay, and you can see, like, viruses are much smaller than bacteria are. Okay, and they're much, much, much smaller than eukaryotic cells are. This is the red blood cell. The red blood cell is literally pretty much the smallest cell in the human body, actually. It doesn't have a nucleus and all this other stuff. It's literally a very small thing already. So you can see how much smaller viruses really are. Okay. So the small size of viruses help. Okay. So I'm gonna try to answer this question for you. How does small size of viruses help researchers detect virus before the invention of the small microscope? Well, we had filters that filter out bacteria. You learned that in the last chapter. If you run a virus through a filter, the virus will run through the filter, and viruses will be in the solution at the bottom of the filter. So if that stuff is still infectious, that means it's not a bacteria that's doing the infecting, because the bacteria will be caught by the filter. Okay. All right. So blah, blah. Okay. So a viron is by definition a completely developed viral particle. Okay. Okay. The nucleic acid, the DNA can be single-stranded, double-stranded. The DNA can be linear or circular. RNA can be single-stranded or double-stranded, and RNA can be linear or circular. Okay, and RNA can also be the plus strand or the minus strand, which is actually really kind of incredible. Okay, okay. The capsid. So viruses have a DNA, okay, or nucleic acid, and a protein coat. The protein coat is known as the capsid proteins. Okay, some viruses, COVID-19 included, have an envelope. Okay, so when they come, so they make a whole bunch of proteins on the cell, and they put the proteins on the cell membrane, and then when they come out of the cell membrane, they take part of the cell membrane, phospholipid bilayer with it. Okay, now it becomes called a viral envelope, the phospholipid layer surrounding the virus that includes the viral proteins taken out of it, and those are the spikes. Okay, so this is a non-envelope virus, so it just has a capsid around it. This is envelope virus, so you can see there is the yellow stuff is the phospholipid bilayer of the cell that it came out of. They made a bunch of proteins that they stuck on the phospholipid bilayer, and that those are will end up being the spikes on the virus. Okay. COVID-19 looks pretty similar to this, except the spikes on it is much longer. Okay. Okay, helical viruses. Well, here's all these things I really have to know. Okay. The taxonomy of viruses. Viruses obviously don't breed with each other, right? They don't, mate or So there's viruses. Um, the species is a group of viruses sharing roughly the same genetic information and the same ecological niche, the host. Okay. Um, so you can see the genus names, order names, etc. Okay. So bacteriophages. Okay. So bacteriophages are viruses that infect bacteria, and so you can see on display what and why they're called bacteriophages. Okay, and remember we named them before we actually really understood what they were. Okay, but you can see there are holes in the bacteria here. Okay, and it looks like the bacteria lawn, right, has been eaten, like bits of it has been eaten, and so bacteriophage. Okay. Okay. All right. So growing animal viruses in the laboratory, in living animals, right? So viruses are not bacteria. We can't grow them by themselves. They need a living cell to infect. And a lot of viruses, we can't even grow them on um, cell-like culture because they're not right for cell culture. Okay, but so we can, infect them in living animals and keep them in living animals. But easier is the actual egg in actual eggs or embryonic eggs, okay, which means they're actually fertile and not your, the eggs that you had for breakfast, okay. And this is preferred because it's simpler, okay. The virus lives inside the egg, right? Um, it doesn't move. You don't need to feed it. You don't need to take care of it. You don't need places for it to run around. It doesn't shit on itself. Like all this stuff makes injecting the virus into the egg very easy and very very safe and cheap. Okay. Your flu vaccine most likely came from chicken eggs and stuff. Of which one they tell you like if you're allergic to like egg white or whatever, you can't take that vaccine because it has some egg white component to it. Okay, that's why. Okay. So there you go. All right. Don't worry about the different types of injections, right? So, in cell cultures, tissues are treated with enzymes to separate cells. The infected cells are detected via the cytopathic effect. Okay, so you can see, okay, these are normal cells. These are transformed cells. Transformed cells are infected by virus. Okay. Usually they do this for viruses that cause cancer, because then you can see very clearly, like if you have abnormal cell growth, like this group of cells is growing faster than that group of cells, then this group of cells is infected by virus. So it's a very fast way to see for cancer viruses. For other viruses, not quite as useful. Okay. Okay. Um, So the cytopathic effect of viruses, you guys can see, this is a huge fusion of many cells. And that's one of the things that happen with viral infections. They end up fusing many cells together a bunch of times and form something called syncytia, um, something like that, okay? Okay, the viral identification, how we identify um, things. I'm going to skip the cytopathic effects. The most important things we do is the serological tests, and I don't think we really we do risklets anymore. The RFLPs, we do PCR a lot more often because it's really easy now and more, it tells us more stuff. Okay. All right. So, okay. So, viral replication, this is definitely something you have to know. Okay. Okay. So for a virus to multiply, it must invade a host cell, and it must hijack the host metabolic machinery. Okay. So there are two types of cycles when it comes to bacteriophages: the lytic cycle and the lysogenic cycle. Make sure you understand both. Okay. Oh. Okay. So here is the lytic cycle. Okay. And we'll up this screen. So.
Virulent phages carry out a simple life cycle called the lytic cycle. The phage attaches to the host cell during the attachment stage and then penetrates the host cell and injects its DNA during the penetration stage. After penetration, the phage causes the host DNA to break into small pieces. The phage then uses the host machinery to synthesize new copies of its DNA. This process is part of the biosynthesis stage. Biosynthesis also involves production of viral proteins. Once biosynthesis is complete, the phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages go on to infect other cells.
Okay. 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 This is the lysogenic cycle, and after they finish talking, I'll explain a little further.
Temperate bacteriophages carry out two types of life cycle, the lytic cycle and the lysogenic cycle. The lytic cycle for temperate bacteriophages is similar to the lytic cycle for virulent phages. The phage attaches to the host cell during the attachment stage, and then penetrates the host cell and injects its DNA during the penetration stage. In temperate bacteriophages, the phage DNA forms a circle, which can either replicate and be transcribed to produce phage components in the lytic cycle, or can proceed to the lysogenic cycle. During the biosynthesis stage of the lytic cycle, the phage DNA directs the host cell to synthesize viral components. The phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages can go on to infect other cells. Recall that temperate phages can proceed either to the lytic cycle or the lysogenic cycle shortly after penetration. In the lysogenic cycle, the phage DNA integrates within the bacterial chromosome by recombination. The inserted phage DNA is called a prophage. Most of the phage proteins are repressed by two repressor proteins that are products of the phage genes. Whenever the bacterium reproduces, the prophage is also copied. The prophage is excised from the host chromosome in a process called induction. Induction can occur spontaneously through recombination or some other genetic event, or through the action of UV light or certain chemicals. At this point, the phage may enter the lytic cycle.
Okay, so, oops, sorry about that. Okay, so here is the lytic cycle, okay? Okay, first of all, overall rule. Viruses want to make new viruses, okay? Second rule, gene theory, okay? Genes want to make new genes, okay? The proteins are not really involved in this, okay? So a tiger is a way for tiger genes to make new tiger genes, okay? Humans are a way for your human DNA to make new human DNA, okay? Viral DNA is a way for viruses are a way for viral DNA to make new viral DNA. The thing that really matters, the actual mechanism, the actual thing that actually gets replicated ultimately is not the proteins that are in the virus, it's the DNA. Okay, it's DNA or RNA, it's the nucleic acid, that's the genetic material. That is what's passed on. So the proteins matter less than the DNA, because with the DNA you can always make new proteins. You can't make new DNA with proteins. Okay. All right. I know that was probably a really kind of somewhat philosophical thing for you guys, but really what boils down to for viruses is DNA or RNA, their genetic information, not so much the protein. Okay? So the virus is trying to make new viral DNA, so we can inject its way into this bacterium. Okay? It's going to undergo biosynthesis. So if I'm going to make new virus particles, I am going to cut up the DNA of the host, because then all the defenses are gone, all the host needs and wants are gone. So the host is no longer making proteins for itself. It is just now a big sack of stuff that the virus can hijack. Okay, so I'm going to build a bunch of stuff. So make a bunch of copies of the DNA. Okay, make a bunch of the proteins, assemble them all together, and eventually I'm going to assemble everything into new virus particles that will eventually burst out of the cell. And those things can go on to infect new viruses. Okay, and those things can now become new viruses and infect new bacteria. And so that's why bacteriophages look like rings of empty material. They just chill all the bacteria in that circle and then move on to the bigger circle and the bigger circle and a bigger circle. So the ring, if you keep culturing, it will grow bigger and bigger and bigger. Okay. All right. Okay. That's one strategy. Okay. Another strategy is simply to hijack the DNA and make it do what you want. So instead of making new viruses, it takes the DNA and it inserts it into the DNA host bacterium. Now the host bacterium doesn't know any better. Okay, it just has phage DNA in it, but it thinks it is itself. So every time it replicates, the virus replicates. The viral DNA also replicates. So remember, I told you the virus is only the DNA. Okay, so every time the virus, the bacteria replicates, it will make more viral DNA. So it's actually replicating. It's just not making the proteins. Okay, so the virus is actually replicating. Okay, occasionally it becomes unhappy and then goes back into the lytic stage and kills the bacteria. Okay, so that's the lysogenic and lytic stage. Okay, so basically what happens is when these phages enter the bacteria, it has to make a choice on whether to go lytic or lysogenic. And what that choice is based on is relatively simple. If the bacteria is growing well, it's more likely to go lysogenic. If the bacteria is not growing well, it's more likely to go lytic. Okay? If the bacteria is all happy and shit, right? So you gotta think from the point of the virus, if the bacteria is growing, then you don't want to interrupt it growing. You just want to grow along with it because bacteria grow pretty quickly on their own. So you can just insert your DNA, and every time the bacteria replicates, you replicate and great, right? So when things are going well, it will go lysogenic. When things are going badly, it will go lytic. If things are going badly for the bacteria, I don't want to stay there. I don't want to hitch my wagon in something that's already failing. I just want to hijack as much of it as I can and just get out. Okay, so that's the difference between the two cycles. Okay. Okay. You can skip the specialized transduction. Okay. You can watch this video on generalized transduction. Pretty sure you guys already understand it, but
in transduction, DNA from the chromosome of one cell is transferred to another cell via a replicating virus. A virus that infects bacteria is called a bacteriophage or phage. There are two types of transduction, generalized transduction and specialized transduction. Generalized transduction, as shown here, is initiated during the Lytic cycle of virulent bacteriophage. The donor DNA can be any fragment of the bacterial chromosome. Specialized transduction, which is shown on a separate animation, is initiated during the lysogenic cycle of a temperate bacteriophage, and the donor DNA is a very specific part of the bacterial chromosome adjacent to the prophage. At the beginning of generalized transduction in bacteria, a bacteriophage attaches to a bacterial cell wall and inserts its DNA. The phage commandeers the bacterial cellular machinery to synthesize the new phage DNA and produce phage proteins. In order to completely convert the bacterium into an efficient virus-producing factory, some of the phage proteins break the bacterial DNA up into small pieces. Normally, phage DNA is coated with phage protein to form new virus particles, but sometimes pieces of bacterial DNA are mistakenly packaged in phage proteins. These sloppily packaged phage particles carry bacterial DNA rather than phage DNA and are called transducing phages. When a transducing phage, composed of phage proteins and bacterial DNA, infects a new host, the phage injects the bacterial DNA into the new bacterial host. The infected bacterium sometimes incorporates this DNA into its own chromosome through recombination, resulting in the transfer of genes from the donor bacterium to the recipient bacterium. In trans
Okay. So how do bacteriophages get nucleotides and amino acids if they don't have any metabolic enzymes? They hijack it from the host, right? Viral cholera actually produces toxins capable of causing cholera only when it's lysogenic. This is actually a really important thing, okay? So what does this mean? It means it only, the virus isn't killing, the virus that causes, sorry, the bacteria causes cholera. Okay, that's what the viral cholera is. But it has to have a phage with it to actually do the cholera, or else it's just a transient host, really, that it's supposed through. Okay? It's the virus that provides the toxin. And the toxin of the virus is something that makes you pump incredible amounts of water into your intestines. And I know this sounds disgusting, but you literally shit yourself to death. Okay? All right. So, blah, blah, blah. Okay. Okay. The virus is attached to the cell membrane. Okay. Okay. So the multiplication of animal viruses. So animal viruses. This is the animal virus. The insertion is pretty much the same. So the virus. So bacteriophages have a hypodermic needle-like structure where they inject the DNA. The animal viruses are slightly different. They're slightly more complicated. The virus fuses to the membrane and causes endocytosis. It has receptors on the surface to cause endocytosis of the virus. And once the virus gets inside, it uncoats, it unravels the protein envelope. Okay.
Viruses are agents capable of infecting cells and reproducing, but it can be argued whether a virus is alive or just a complex group of molecules, since it requires a host cell in order to reproduce. Whether they are alive or not, they're able to co-opt the host cell's machinery in order to do the bidding of the virus, the replication of the genetic material of the virus, as well as the structural elements that package and protect the genetic materials and enable the virus to infect new host cells. There are many different types of viruses infecting a large range of host cells. They all follow the same general life cycle, with variations in the details of how each accomplishes its replication.
So
Animal viruses vary based on the type of nucleic acid they possess, as well as whether they are naked or enveloped. Each variance results in different types of life cycles. A naked and an enveloped virus will attach to and penetrate a host differently. Animal viruses without an envelope are referred to as naked viruses. They bind to the surface of the host cell and inject their DNA into the host cell in a manner similar to bacteriophages.
naked viruses are rare. Okay.
Some envelope viruses infect their host by binding to receptors on the host cell. The envelope of the virus then merges with the host membrane and the capsid enters the cell. After entry, the capsid opens and releases the viral genetic material into the cytoplasm of the cell. Upon attachment, some envelope viruses infect their host by inducing phagocytosis, in which the host cell envelops the viral envelope.
This is a little more, these two are common. This one's a little bit more common.
And absorbs the virus into its cytoplasm. When the virus has entered the cell, the outer and inner part of the envelope merge together, and the capsid is released into the cytoplasm. At this point, the capsid opens up and releases the viral genetic material into the host cell cytoplasm. The type of nucleic acid in an animal virus will determine how the viral nucleic acid and proteins are synthesized. When the single-stranded DNA genome of a parvovirus enters a host cell and invades its nucleus, a complementary strand is produced for the single-stranded viral DNA. This DNA is then replicated normally. Messenger RNA is transcribed and transported into the cytoplasm, where viral capsomere proteins are produced. At some point after synthesis has begun, the capsomere proteins enter the nucleus of the host, and the virions containing the original single-stranded DNA are assembled. In a virus with double-stranded DNA in its genome, the DNA also migrates to the nucleus, where it's replicated by the host enzymes in the same way that the host's DNA is normally replicated. Messenger RNA is made from the viral DNA in the nucleus, and viral capsomere proteins are made in the cytoplasm from the messenger RNA. The capsomere proteins enter the nucleus, and the virions are assembled in the host nucleus. After assembly, the virions accumulate in the cytoplasm and prepare for release from the host cell. Some viruses have a single-stranded RNA genome that can act directly as messenger RNA. This strand is called the positive strand or sense strand RNA. This messenger RNA is read by host ribosomes to make viral proteins. The virus carries a unique RNA polymerase that makes a complementary negative strand, also called an antisense strand, from the original positive strand. There is no known animal equivalent of this RNA-dependent polymerase. The negative strand RNA can act as a template for more positive strand RNA. Positive strand single-stranded RNA viruses are assembled in the cytoplasm of the host cell when the RNA and the proteins have all been produced. Viruses with single-stranded negative sense RNA are in a special situation. Their RNA will not act as messenger RNA until it's been transcribed into the positive sense strand complement of the original negative antisense strand RNA. To accomplish this feat, the viruses carry their own RNA-dependent RNA polymerase, which transcribes the RNA into the positive strand. When the positive strand has been produced, it acts as the template for both protein synthesis and more negative strand RNA. When the virion is packaged, it must contain the negative strand RNA in addition to copies of the RNA-dependent RNA polymerase. When double-stranded RNA viruses infect a host cell, the positive sense strand of the RNA acts as a messenger RNA for protein production. One of the proteins coded is an RNA polymerase that uses the negative strand to make more positive strand RNA. The positive strand also acts as a template to make more negative strand RNA. The two strands come together and are packaged along with the polymerase in the virion. Retroviruses make up a special class of positive strand RNA viruses. When a retrovirus infects a cell, its positive strand RNA is reverse transcribed into a DNA negative strand by a viral reverse transcriptase. This negative strand DNA is further transcribed to double-stranded DNA, which serves as a template for the viral RNA genome. This positive strand RNA also serves as the template for viral protein synthesis. The reverse transcriptase is packaged with the mature virion, as it's essential for the successful reproduction of the virus. Just like the beginning of the life cycle, naked viruses differ from envelope viruses in how they assemble and release. Naked viruses generally leave their host cell by accumulating to such numbers that the cell lyses, spilling virions into the extracellular medium, free to infect new hosts. This kills the host cell.
Okay, again, this is rather rare, okay? Most animal viruses are enveloped, and you'll see that how that plays actually an important role in things.
And generally causes inflammation and infection in the tissue where the host cell is found. Enveloped viruses leave the host cell by merging with one of the cell's membranes, the nuclear membrane, the endoplasmic reticulum, or the cytoplasmic membrane. During synthesis, some viral glycoproteins are embedded in cellular membranes, and these glycoproteins act as a recognition system for the viral capsid. The cell membrane surrounds the capsid and forms a bud on the outer surface of the cell, which pinches off and forms a protective envelope for the virus, which is then free to infect other host cells. An advantage of the envelope is that the virus does not need to break the cell to be released, so the cell can stay alive longer while it continues to produce and bud off more viruses. The type of nucleic acid, along with the presence or absence of an envelope, will determine the type of life cycle a given animal virus will have.
All right. So again, so the viral envelope is actually very important because, like, you see that the proteins are made by the virus, but they're in the phospholipid bilayer, and these are going to be the attachment sites for the next infection. Okay. All right. So, okay. Okay. So we just went over all this in the video. Okay. We'll skip Adenoviridae. Really cool. Pox viruses. Okay. Herpes virus. Okay. Herpes. There's a whole bunch of them, like HHV5. There's a disease called CMV, cytomegalovirus. Herpes 1 and 2 is called herpes simplex virus. They cause cold sores or genital sores. Herpes simplex virus 1 is above the waist. Type 2 is below the waist. Okay. Papillomaviruses. Okay. Papillomaviruses cause cancer. Okay. So I'm sure you heard of HPV. Okay. That causes cervical cancer in females. Which is why there's a vaccine, and if you're a sexually active female, I highly encourage you to take the vaccine. Okay. So you should know the difference between the plus strand and the minus strand. Okay. Let's skip all this. Okay. Bacteriophage, Bacteria pico, small RNA virus. Okay. What's called poliovirus, rhinovirus, rhino nose, right? So causes about 50% of the common colds. Okay. Cultivation, Rhabdoviridae causes rabies. Okay. Like I said before, rabies virus. Actually, I don't tell you guys until another class. Rabies virus is one of the scariest viruses ever. And it's a really simple virus actually. Okay. Rotaviruses cause mild respiratory infections and gastroenteritis. It also basically causes massive amounts of diarrhea. Okay. The ones that I want to really kind of focus on are the Retroviridae, okay? Because that's what the HIV virus actually is. Okay. The Retroviridae, go back, right? So retroviruses go from RNA to DNA. Usually it's DNA, RNA, protein. This retrovirus has an enzyme called reverse transcriptase. Reverse transcriptase takes RNA and makes a DNA copy. So when the virus gets inserted in, it inserts viral RNA, but it gets immediately made into viral DNA and inserted into the genome. Okay, which is why, like, once that cell has been infected, it doesn't die, or it just inserts it into the cell. When it inserts the virus into the cell, it's called a provirus. Okay, and now the cell will function and just make, just like provirus. Okay, the cell will stay alive and just make virus particles that eventually bud out of the cell. Okay, and will do this for a very long time until it dies. Okay. Okay. All right. Yeah. The next thing I want to make sure you guys understand are oncogenes and transformed cells. Okay. A oncogene okay. So sarcomas are cancer of connective tissue. Adenocarcinomas are glandular epithelial tissue. Okay. Oncogenes, by definition, are transform normal cells into cancer cells. Oncology is the study of cancer. So oncogenes are genes that causes cancer. Okay, oncogenic viruses become integrated into the host cell DNA and induce tumors. Remember, rule number one of virus is to make more virus, in particular the DNA. If I'm a DNA virus, I insert myself into the host cell. If I get the host cell to make more host cells, then I have more cells infected. Okay? The virus isn't trying to kill you. Like, killing you is a byproduct of what it's trying to do, which is make more virus. Okay? Okay. So these are the DNA oncogenic viruses. Okay. Human papillomavirus, hepatitis B, are the main ones that you have to know. Okay. The Epstein-Barr virus. All right. The same thing for RNA oncogenic viruses. The only type of RNA oncogenic viruses are Retroviridae, really, because they transfer into DNA. Now it's DNA, and the DNA wants to make more DNA, and therefore it's easy to just make the cell grow into more cells. And every time you grow a new cell, you make a copy of the viral DNA, and so the virus DNA replicates. Okay, provide an example of a latent viral infection. Differentiate persistent viral infections from latent viral infection. This is definitely an important concept. 1314 is definitely an important concept. Okay, a latent virus remains asymptomatic host cell for long periods. Okay, let's talk about cold sores for a second. Okay, if you're taking anatomy, you know you have a nerve called the trigeminal nerve. Okay, one of the functions of the trigeminal nerve is sensory perception of the face. Okay, the virus, the cold sore virus, actually stays in the ganglion associated with the trigeminal nerve. Okay, so trigeminal nerve ganglion. It will stay there. It will do nothing there. It will rest there, and it will just stay out of the way, and therefore the white blood cells can't see it. Okay, occasionally it will reactivate and cause a cold sore and travel down the nerve. That's why it's fixed. Okay. These cold sore outbreaks are usually due to changes in immunity, and they're changes in immunity due to stress. So when you're stressed out, you have more frequent cold sore outbreaks. Shingles. Hopefully, you guys mostly won't ever get shingles because you guys are never had chickenpox. Okay, chickenpox never goes away. Okay, if you had chickenpox, it never goes away. It hides. Okay, in one of the dorsal root ganglions in the body. Okay, the dorsal root ganglions again innervate parts of the body, but they innervate particular parts, right? So there's a pathway, and occasionally it'll activate, and when it activates, it's shingles. So when you ever look at someone with shingles, you'll see like a bright red line, like where that nerve is, and that's what becomes inflamed. It's incredibly painful, right? But it's all on one nerve, and you can see the inflammation of the nerve. Okay. Latent viral infections are dormant. Okay, a persistent viral infection is not dormant. It's persistent. It keeps going and going and going until it freaking kills you. Okay. So the primary effect of latent is that there's no symptoms, right? And then here's the shingles, leukemia, etc., etc. Okay. Viruses, virons, prions. Okay. Virons are short naked pieces of RNA. Okay. They're a loop. They're not really a short piece. They're a loop of RNA. That loop of RNA somehow infects the cell and causes that cell to, instead of making RNA for other stuff, it just makes short loops of RNA, the viroid all over again until it makes so much viroid that it destroys the cell, and the cell just kind of leaks out and then infects other cells with viroids. Okay. All right. Okay. So that's the only thing you have to know about viroids. Prions are infectious proteins. Okay. So let me show you what prions actually do.
All mammalian cells contain a gene that codes for the primary sequence of amino acids for the prion protein PRP. Normally, PRP folds into a functional form with several alpha helices called cellular PRP. The normal function of the prion protein is not well understood, but it's believed to be important in normal synaptic development and function, and it may be involved in stabilizing the structure of the synapses and in establishing memory. The prion protein is also capable of folding into a form with beta-pleated sheets. Prions do not reproduce like bacteria or viruses. Instead, infectious prions convert normal prions into more infectious prions by folding them into beta-pleated sheets. Thus, the number of infectious prions is increased, but by conversion rather than traditional reproduction.
Okay. The beta-pleated sheets is important. The beta-pleated sheets are incredibly stable. They're very hard to break. In fact, they don't. These guys are so super stable. They don't break at all. You cook them, you heat them. Okay. And now you see there's no more of the regular stuff. So
Prion proteins in the infectious conformation can group into multimers, which are very stable and resistant to protease. Over time, these multimers probably lead to some of the damage seen in prion-infected tissues.
Okay. So now there's no more of these guys. The cell will sense there's no more of these guys, and it will make more of these normal PRP proteins. The prion will bind to those and make more prion, and on and on and on, until basically all you have is a whole bunch of these prions, and they will just kill the cell. And as the cell, when the cell dies, it will burst these prions out, and they will infect other cells and do the same thing. Okay, so there's no genetic information involved in this cycle. There's just prions. Okay. All right. So let me explain all these guys. Okay. So the proper term for mad cow disease is bovine spongiform encephalitis, or encephalopathy. Okay. Bovine cow, spongiform, sponge-like structure. It literally pops holes in the cow brain. Okay. In humans, it's something called Kuru. Another symptom, another disease is the Creutzfeldt-Jakob disease. Okay. In sheep, it's called scrapie. Okay, because literally, sheep go crazy and scrape themselves against like enclosures until they bleed out, like bleed profusely, and eventually die and crap. Okay. All right. So I'm gonna explain what all this stuff are. Okay, mad cow disease. I'm sure you guys have heard of before, but here's what happened. Okay, when you take a cow, you want to use a cow as much as you can. So you'll take a cow, you'll kill the cow, you'll sell off the steak. Whenever you don't make a steak, you turn it into a hamburger, and then you have still the leftovers, the bones, okay, the nervous system. Like people don't eat that many cow brains anymore. The cow brains, the hooves, the spinal cord, all this, stuff that's left over. Okay, so what do you do with it? You can throw it away, but that's a waste of stuff. So what do you do with it? You cook it. Okay, so the farmers decided to cook it and feed it to more cows, because if you feed cows protein, they'll grow faster. Okay, and this stuff they're cooking is proteins, and the reason is pretty straightforward, right? So I'm gonna cook the shit out of the cow. I'm not gonna worry, like it's not gonna transmit any virus or bacteria because I'm cooking the shit out of it. Like you really, I mean, by cooking the shit out of it, they don't. It's not just like well done. It's like powder. And you know, this should have prevented diseases from spreading. What they didn't realize was prions are. First of all, they didn't know anything about prions back in the day. This stuff was so infectious and so resistant. Remember I said the beta plate sheets don't get destroyed and stuff? They're resistant to even that level of cooking. So it got spread to the cows very quickly. Okay, from one cow to another cow. Okay, and that's how prions spread, from feeding cows, other cows. So as soon as they figure all this out, the cure is simple. Stop feeding cows other cows. And so now there's regulations for doing that, and now that will spread again. Okay. In humans, okay, there's a disease called Kuru. Okay, Kuru was only found in one area of New Zealand. Okay, these tribes were pretty poor, and they had no source of protein. Okay, they didn't have any farm animals, they didn't have chickens and stuff like that. Like they didn't have any source of protein for their diet. Okay, there was no big game. So what they did was they kind of survived by having rituals. And one of the rituals was when someone died, they would have a feast and engage in ritual cannibalism. They would eat the dead person. Okay, and when they ate the dead person, they would obviously eat the brains, and this is how Kuru would transfer from person to person. Okay, if you can go on YouTube and you can look up Kuru, and you can see like what the symptoms of Kuru are, but basically, you see these women who are like stumbling around, like completely uncoordinated. And the reason why it affects women is simple. If you observe the ritual, the men ate first. The men ate first means they ate all the meat, they ate all the muscle, they left behind basically the spinal cord, the brain, the organs that the men didn't want to eat, and that's what the females ate. So the disease is much more rampant in females. Okay. All right. Sounds pretty nice. Okay. All right. Hopefully that chapter made sense. I will see you guys for chapter 14, which I promise is a little easier. I know these last two chapters were difficult. Morning.